Welcome to the fifth instalment of the OpenKMI Modelling Initiative webinar series. So today's topic is modelling non-steady state kinetics in the brain. We will hear presentations from Evan Morris from Yale University and myself. Your panellist for today is Julie Price from Harvard University and Mass General Hospital. After the presentations, Julie will moderate a discussion with the presenters. She will also be inviting questions from the audience, which you can ask by raising your virtual hand or by submitting your questions through the chat function. So my name is Steve Meikle, and I have the pleasure of introducing our first speaker. So Evan Morris is Professor of Radiology and Biomedical Imaging at Yale University. He's been working in PET for 30 years. Evan received his PhD from Case Western Reserve University in 1991, then undertook postdoc positions at MGH and NIH before moving to Indiana and Purdue and finally to Yale with an overlapping time at in vitro. Evan specialises in using kinetic modelling and image processing to extract physiological information from dynamic PET images. Much of Evan's work is focused on imaging the dopamine and opioid systems in cigarette smokers and alcohol drinkers. For many years, he has offered a popular course at Yale, Imaging Drugs in the Brain. He's delighted to see the growing interest in and enhancements to LPNT-PET, the model that we will discuss today. The title of his presentation is Mapping Non-Steady State Dopamine Kinetics with PET, a Historical Perspective. Over to you, Evan.
Okay, thank you, Steve. Okay, I'm going to jump right in, and I'm going to hope that everyone can see my screen and see that the title, as Steve said, is a historical perspective. So how did we get where we are with regard to non-steady modeling of dopamine signals in PET? And first we have to try to define the problem a bit. So it's best defined graphically. Given this time activity curve on the top, we're looking for this dopamine pulse, which explains any dips in the time activity curve. And I would say that, to put it another way, we have to find the dopamine activation in the noisy time activity curve, and it's actually a challenging model fitting problem. So to find the brief pulse of dopamine, we want the pulse that alters the theoretical trajectory of tracer uptake to best fit the PET data, and we want to ultimately do it at every voxel, and we want to do it without knowing precisely when the dopamine response starts or even if it exists. So that's the problem that I am interested in generally. Let's just dig in a moment and define some terms. So again, the same cartoons. You could say that the first half of this time activity curve, which is tracer uptake, represents the system at steady state. And so the dopamine, which is what I'm interested in, the endogenous dopamine, is not changing. That's what I mean by steady state. On the other hand, the second half of the curve reflects some non-steady behavior, and we're speculating that there's a transient dopamine effect in the case of addiction and response to drugs that explains that behavior that we see above. Now, to describe a system that's not at steady state, one might resort to converting a model with time invariant parameters to a model with time varying parameters. That is to say, for example, K3 should be represented as K3 of T. That's the definition of the problem as far as we need to define it, but I'm getting ahead of myself a little bit. So let's consider, here's my outline. How did we get to the model I'm going to talk about? What can we do with it? What are the challenges that remain? And what are some of the latest innovations that I find particularly exciting? So how did we get there? Well, it took a lot of people and a lot of work, but we can sort of date progress back to about 1983 when there was a chemistry innovation. And that innovation was the introduction of a tracer for dopamine receptors. And not surprisingly, many of you, I'm sure, know this paper. When you have a tracer for dopamine receptors, you get an image of dopamine receptors. Now, at this point in 1983, it was not quantitative, but I'm going to come back to that in a moment. But already I have to make my first digression. And my first digression is, oops, if I can get to the right slide, I just learned this little tidbit of PET lore. So although, and I'm calling this my historical note, although it is true that that paper, which many of you recognize by Wagner et al. From 1983, appeared in Science, it's a less known fact that 1 week before that, this was the cover of Nature. It looks awfully similar. So are they the same? Which is the first image of dopamine receptors? Turns out that what's on the left on September 22nd was the first dopamine receptor image, and what was on the right in Nature the week before it was the first image of aromatic amino acid decarboxylase. That is a PET image made with F-DOPA. And to drill down even deeper, one is Henry Wagner's brain and the other is Steve Garnett's brain. So one is the first dopamine receptor image, but you could argue, and as they did, the first dopamine visualized in the human brain came the week before. Okay, now back to my story. So the year after those two papers, Mark Minton introduced a quantitative model, which I'm calling a model of dopamine receptors. You'll see in a moment the distinctions I want to make. And thanks to that model, which is basically mass balances that encode the comings and goings of the tracer, you can estimate receptors, and one of the parameters that we estimated was BP, or binding potential, which is effectively the density of those receptors. Now, a few years later came the first model of bad endogenous dopamine, or the bad, wicked witch of the endogenous dopamine West. Now, why do I say it's the bad dopamine? Because in this paper by Jean Logan, who introduced a model which included a state variable for dopamine, she was asking a particular question, which is represented graphically here. Here, if you can see my cursor, for different strengths and durations of dopamine pulses, how much effect is there on the estimate of dopamine receptors, or as the arrow indicates, how much alteration of the Logan plot is there? Now, the reason it's considered a model of bad dopamine is because the real question at hand there was how much of a dopamine pulse is tolerable before you start to bias the estimate of the thing that they cared about, dopamine receptors. Well, you know where I'm going already. A few years later, we jumped in with what I would call a model of the good endogenous dopamine. Why? Because we were introducing a similar model with differential equations to encode a state variable for dopamine and a state variable, the tracer. But what we cared about was maximizing the effect of dopamine. And what the question we were asking with a similar model was, at what time do you place the task and for what duration to optimize your chances of detecting dopamine? So we've gone from a dopamine receptor model to a dopamine model where we want to measure dopamine. Now, shortly thereafter, a few years after, I should say, Nat Alpert introduced what I will call a linearized reference tissue model of dopamine, moving much closer to a model that is practicable. The previous model had differential equations and required nonlinear fitting. It's slow. But this model, thanks to Nat's linearization, his innovation, produced linear fits and much faster fitting, so the ability to estimate or fit these time activity curves at the voxel level. And it looks like this. The linearization is familiar. It produces an integral equation. And the last term contains a time varying term to encode some dopamine transient. And you can see in this circle on the left below that there's a non-steady behavior in the time activity curve that Nat was able to describe. He could still estimate binding potential, number of receptors, but the really interesting thing here is this measure of gamma, the strength of the dopamine response, which was an exponential. And if you look closely, you can see that in the striatum, the error in that estimate of the strength gamma is low and the t-statistic is large. And there was a significant effect in parts of the striatum that had to be described by a dopamine transient. Now, we jumped back into the fray a few years later, and I would call, in distinction to Nat's model of dopamine, this was a model of dopamine timing. And by that, the distinction I'm making here is that we had, if you see the red, the blue, and the green dots, we allowed for explicit parameters to describe timing of that dopamine, which had heretofore been fixed. So with this model, which still has the shortcoming of not being linearized and requiring nonlinear fitting, we did introduce the ability to estimate the timing, the beginning, the peak, and the return to baseline of the desired dopamine signal. But then a number of years after that, Mark Normandin, in his PhD thesis, introduced a linearization of that dopamine timing model, which could estimate dopamine timing without constraining it to a particular predetermined moment. And he relied on yet two more innovations, one which had been circulating in the PET space, basis functions, to accomplish his linearization, and model selection. And it looks like this. The basis functions I'll come back to, they allow us to linearize this otherwise nonlinear term, which represents the dopamine function h of u over time. But the model comparison innovation, which was the F statistic, which is a fractional improvement in the sum of squares by going to the more complicated model on the second row, which includes a dopamine term, is a way of asking, do we have a statistical basis for saying that the larger model, which includes a dopamine term, is justified? So that was the second innovation that Mark introduced. And then our group, thanks to Sujin Kim, extended this model to voxels. So at this point, we're able, with yet one more innovation, because when you go to voxels and images, you have to worry about multiple correction for multiple comparison. Sujin introduced a cluster size threshold. So it looks a little bit like this. The top panel on the very left is the F values to determine which voxels require a more complicated model. But what's on the bottom left shows you the distribution of cluster sizes that happen by chance. So, of course, the most common cluster of apparent activation is one voxel, but it happens by chance so often that we have to threshold it out. And in this case, what we're showing in Sujin's paper is that we were able, even after these two criteria that reject possible false positives, we were able to find clusters of distinct location and distinct timing. So what could we actually do with that? It's all mathematically kind of interesting, but what did we actually do with it? Well, first in animals, we used it in 2008 to ask, what is the dopamine response to alcohol in anesthetized rats? And we did have a comparison with microdialysis. So what I'm showing you here is after drug, there is this drug being alcohol, transient response in blue of the dopamine signal, but the red is microdialysis, which is in a sense the gold standard, at least it's extracellular dopamine. So you can see that in the drug case, the microdialysis and the model estimate of the dopamine curve seem to agree pretty well, but in the control case, the model doesn't find anything, and effectively the microdialysis doesn't find anything, except perhaps a small response to cold raclopride. Okay, but really that was just a setup, because our case was anesthetized animals, and in the a little bit later, this was used in a much more exciting way, which you're going to hear about shortly, but it looks like this for starters. Steve and his team in Australia were able to use the same model on data from awake rats. And I know we're going to hear more about it. But you can see on the right in the panel that non-steady behavior in the top curve, which is the striatum, requires a transient in dopamine to explain its behavior. Okay, we also used this model in people, and here's the most interesting thing we found was a sex difference. But the really cool thing is that we had people smoking cigarettes in the PET scanner. That's our drug delivery. And we're asking where and how much and for how long do smokers respond? Does their dopamine system respond? And the first thing we found that was when we used the appropriate model with our transient term for dopamine, there were differences in location of activation between men and women. So that was interesting. Again, responding to their own cigarette that they smoke in the PET scanner. And there were differences in the timing of those responses, blue being men and pink being women in this particular curve in that cluster. But if you have time, let me go back for a moment and build up to this next slide. If you have dopamine concentration minute by minute or frame by frame at every voxel, I would argue that you have a movie of dopamine. And here are two examples, and one is already running on the bottom. I hope it's running on your screen too. If you watch closely, now they're both running. The bottom row is 4 slices in a man's brain while he's smoking, and there's the dopamine activation inside the yellow oval. And then slightly later and for a more prolonged period is the women's activation. So it'll go around one more time. Ventral striatum, early is the response of men, slightly later and for more prolonged is the response of this woman, but there was some similarities to other women. Okay, so now the question is, that's all well and good. There was some excitement about what we could do with it, which we couldn't do before, but there were still challenges which are now being addressed. That's what I want to talk about in my remaining time. So what are those challenges? They always come down to 2 things, I would say, in model fitting: false positives and false negatives. And in this particular display of a simulation, which was from Heather Liu's thesis, the left shows you a ventral striatum phantom, where all the white voxels are truly activated and the black enclosed in the border is truly not activated. It's the null. But unfortunately, when you look to the right, the detected activity is quite sporadic. All the blue areas are false negatives and the red dots are false positives. So why do we see these things, and what can we do to address them? Why false positives? Typically, they're the result of overfitting the noise in the data. And why false negatives? Typically, I believe they're overly strict rejection criteria. And remember, I've given you two rejection criteria. One is at the modeling level. The F statistic says we need the model with dopamine. And one is at the image level. It says, no, I can't accept that cluster size. It's so small, it's likely to have happened by chance. So what have other people done with the most recent innovations, really in the last 4 or 5 years, to address the false positive and false negative issues? Well, first, some people have tried to address reducing the noise in the data. And in this particular case, also from Steve's group, first authored by Yorgos and Hellas, they used neural nets to do some denoising of the time activity curves. And when they did that, they got more consistent, reliable fits to the data. So that's related to reducing false positives. And there has also been quite a number of groups working. There are other groups working on that, reducing the noise, but there have been a few innovations that I want to point out on the modifying or improving the rejection criteria. And one I want to give credit to my former student, Heather Liu. In her case, she looked at the model comparison metric. Remember, that's the first rejection criterion, the F statistic. And she asked a question, and we found an interesting phenomenon. And it was this. Recall that one of the other innovations to achieve linearization of the model is to allow the fitting routine to only select from a limited number of predetermined basis functions. That's the basis function innovation. But unfortunately, what Heather observed was that as the number of basis functions, the more possible different shapes of dopamine curve that we were allowing the model to pick from in a lookup table, you could say, the more the false positive rate. That doesn't make sense. The false positive rate is on null data, shouldn't have anything to do with basis functions. So we realized, that we had to make a correction, and that correction was to the F statistic, or to the degrees of freedom, and we called this effective number of parameters, because the basis functions that we allowed the model to select from don't span all of parameter space. And so it's not right to penalize the weighted sum of squares in the F statistic by the full number of parameters in each of the basis functions. And you can see in this image that as you go from left to right, whether you're on the top row or the second row of the image, the number of basis functions does no longer affects the false positive rate. There are null voxels everywhere except in the very middle. Okay, so that was working on fixing the rejection criteria. Connor Bevington and Vesna Sosi and their team at University of British Columbia was also at the same time making other corrections to the F statistic as a rejection criterion. And what they observed was that the F values that are calculated when you ask the question, can I explain my time activity curve with MRTM, the simple model, or do I have to move to the more complicated, larger model with the dopamine curve? Those F values were not distributed properly as an F distribution. And a lot of them were less than the threshold, and they hypothesized that these F values were improperly biased downward because of partial volume error, typically around the edges of the detected clusters. And when they managed to correct their distributions, they found that they could recover. So look at the orange arrow pointing to an example of a cluster in orange in the middle row. Once they fixed their F distribution, they fixed their rejection criterion, they were able to detect clusters that we were not previously able to detect with our model LPNT-PET alone. And so finally, I think I have another minute to say that hot off the presses is yet another innovation from the British Columbia group, which was presented in Brisbane this summer, where they're taking a different approach to improving detection. Okay, so improving sensitivity, reducing false negatives. And they call this, I'm sorry, my mouse is oversensitive apparently. They call this residual analysis, and it looks like this. Their approach is, if you look in the top left time activity curve, there's a comparison there between a time activity curve with a little bit of that non-steady behavior that I've been showing you, and one right above it, which is a canonical time activity curve for uptake of a tracer in the striatum, let's say. But what they found is that they had good luck and high sensitivity to detecting the effect of a dopamine transient if they calculated the residuals. So in green right below are the residuals between the canonical curve and their observed time activity curve, and then they fit that residual curve to a simple model, and they ask the question, do I need a simple step function convolved with some curve, P of t, to best describe my residuals? And if so, is my beta, the strength of that convolved step function which describes the residuals, significant? And like others, they show that their improvement, which is the phantom is in the middle panel, and on the bottom right is their ability and their sensitivity to detect those clusters. And then what people are starting to call vintage LPNT-PET is on the top. It's not as successful. So I know that it's time for me to move from the stage almost, when everything that I do is starting to be called vintage. And I'll just say that these are just a few of the recent innovations that actually makes me quite happy that people are taking an interest and have new uses for our model. And I want to credit some of them. They're in the Australian group. They were in our group. There's a group in University of British Columbia, and there's a group in Orsay at the bottom. And I think we're going to hear a little bit more about the last few examples. So I just have to wrap up by saying thank you to all these people. A lot of people got a PhD on this work. So it wasn't for naught. And I think our agreement is that we'll take questions at the end of Steve's talk. So now I'm going to stop sharing and give the floor to Steve. Okay, so it's my pleasure to introduce Steve Meikle. Steve is a professor of medical imaging physics at the University of Sydney. He received his PhD from the University of New South Wales in 1995. Steve began his career as a medical physicist at Royal Prince Alfred Hospital in Sydney, spent a year at UCLA as a visiting scientist, and then took a postdoc position at the MRC cyclotron unit at Hammersmith Hospital in London before joining the University of Sydney in 2004. His research aims to develop advanced imaging technologies and quantitative data analysis methods for clinical and preclinical radiotracer studies. These studies are intended to elucidate the molecular mechanisms regulating brain function in health and disease. Today, Steve's presentation will be modeling non-steady state kinetics in the brain, a Bayesian approach. And I just want to end my part with a personal note. I have to say that Steve and his colleagues are absolutely delightful collaborators. But finding a time to meet by Zoom is a non-steady and highly ill-posed problem of its own.
It sure is. Thank you, Evan, for that really kind introduction. and the feelings are reciprocated, Evan. We're having a lot of fun with this collaboration. So Evan introduced the problem that we're looking at here, which is given this set of noisy dynamic PET data of a receptor binding ligand, typically a dopamine receptor binding ligand, find this. So we're trying to find the dopamine signal in amongst some very noisy PET data. Now what I want to do is reframe the problem a little and look at it from the perspective of a Bayesian statistician. Not that I'm a Bayesian statistician, I've still got my L plates on, but a Bayesian would look at the problem this way. He or she would say, okay, so we have a prior belief, which is often expressed as a model of what we expect might happen. And so that would give us a probability density on our model parameters. We call that the prior. So it's P of theta. So theta is our vector of model parameters. So that's our prior belief. Then we do our experiment, take some observations, and that gives us another conditional probability. Now we've got the likelihood function, which expresses the likelihood that we get a particular distribution of PET measurements given some underlying model, our vector of model parameters. And then after having performed the experiment, we update our prediction model based on the newly observed data. And that gives us another probability distribution. So we now have the conditional probability that our vector of model parameters are true based on the observed data. So that's our posterior density function. And most of my talk is going to focus on this function, the posterior. I'm going to try and explain why that's a useful entity to estimate, and how we go about estimating it and how those methods perform. But first, we need to take a look at how these different probability distributions are related to each other. And to do that, we turn to the very familiar Bayes rule, which was devised by the 18th century statistician Thomas Bayes. And so Bayes' rule says that the posterior probability, shown in blue on the left here, is related to the other distributions in the way shown here. So it's a very simple relationship. You've got the likelihood function multiplied by the prior, and it's all normalized by the marginal likelihood. So this is the probability density integrated across all the model parameters. So, so then once we have this posterior probability density, we can perform Bayesian statistical inference on that posterior. So we can analyze our expectations based on the data we've measured. But the question is, how do we calculate or estimate this posterior density function? So there are three main methods, and I'm going to take you through those methods. Probably two very well-known methods in the statistics community, but I'm also going to just touch briefly on deep learning. So the most common and arguably the gold standard method is Markov chain Monte Carlo. So this is basically a random walk algorithm. And I assume you can see my cursor here, but the idea is to take an initial sample from the prior distribution. So you might have a flat uniform prior for your particular model parameter. You take a random sample and you take another sample and you compare those two samples. And you compare them to decide whether or not to accept this new sample, and it's based on a ratio of probabilities. So you're trying to determine the relative likelihood that this new sample is more likely to actually have come from the posterior distribution. So you're doing a probabilistic testing of whether to accept or reject that sample. Excuse me. So this algorithm takes quite some time to settle into an equilibrium, but once it does settle into an equilibrium, the accepted samples then become samples of your posterior distribution, and it provides it converges to an accurate estimate of your posterior probability density function. And this is what it looks like in practice. This is a figure from a paper by Eras et al., the Lyon group. I think it shows quite nicely just the idea of the generation of this Markov chain. So you can see that the burning phase there is shown in grey as the algorithm is settling down to I won't use the word steady state just to avoid confusion here, but it's settling down into an equilibrium. And then all the samples from then on that are accepted in blue contribute to this histogram shown on the far right, which is your posterior density function. So MCMC has a couple of important advantages. Firstly, it provides an asymptotically unbiased estimate of the posterior, and convergence is guaranteed. If you iterate for long enough, it will converge to that estimate. The drawbacks of this method are that in practice, convergence is actually very slow to achieve. And in fact, we can't be certain when we've reached convergence. Secondly, it requires an analytical expression for the likelihood function. So that was one of the entities that we needed to make that acceptance rejection test. And finally, it's a difficult algorithm to parallelize. So it's just computationally inefficient and very slow to perform this, particularly if you wanted to do it on a voxel by voxel basis. So another method that can create or estimate our posterior distribution is approximate Bayesian computation, or ABC. And so I'm just showing, again, schematically the three main probability distributions, but now I've got the posterior that we're trying to estimate over here on the left. And you can see the relationship between these expressed by Bayes' rule. So with ABC, what we do is take a random sample from the prior distribution. So there'll be a separate prior distribution for each of your model parameters. You take a random sample from each, and then you simulate your time activity curve. So you're actually simulating the likelihood function. You're putting your samples of the prior model parameters, sample from the priors of the model parameters into this simulation tool. So this is something that we as kinetic modelers are very used to doing. We do it all the time. So instead of trying to calculate the likelihood function analytically, we're simulating it. And then the next step is to look at the distance between our simulated curve and our actual observed curve. So we can look at the residuals. We can use an L1 norm or an L2 norm to look at the distance between those two entities. And we also have this other important parameter, which is a tolerance parameter. I won't go into all the details of this method, but this tolerance tells us the likelihood of accepting or rejecting our simulated curve with the selected parameter samples. If we accept, then that sample goes in to make up our part of our posterior probability density. So again, it's generating a histogram based on your accepted samples from the prior. And this is what this looks like in practice. This is some simulated data from my colleague, Clara Grazian. And it's in this case, she was simulating a one tissue compartment model. All of the grey curves here are all the trials, all the simulations of the time activity curve based on samples from the prior distribution for K1 and K2. The red curve is the actual noisy observed or simulated PET data, and the blue curves are the ones that she accepted with the tolerance that she was using in this case. And this is an example of what those posterior distributions look like once you've estimated them. The red vertical bars here are the true values, as these are simulations, so we know what K1 and K2 are. And this graph was just to show that if you set that tolerance to be too large, initially your posterior distribution is going to look fairly flat. It's actually going to look quite a lot like the prior. In this case, she started with a flat uniform prior. But once you get that tolerance down enough, the posterior distribution converges to something that's much more concentrated around the true value. And there are ways to know when you have reached the right, achieved the right level of the error rate to give you an accurate posterior distribution. So the advantages of this method is that it doesn't require an analytical expression for the likelihood function. It's easily parallelized for efficient computation. The disadvantage, however, is that it is an approximation of the posterior distribution, whereas MCMC is guaranteed to converge to an unbiased estimate. And I should say that provided you take care with this method and select both the priors and the tolerance appropriately, you get a very good estimate of the posterior distribution. Okay, so that's a brief introduction to two of the common methods for estimating the posterior. So how do we apply this to the problem that Evan very eloquently outlined? Well, so here is the now well-known vintage LPNT PET model. So we've got our data on the left with a model fit using the basis function fitting method. And this is our linearized operational equation on the right. So as Evan said, it's this last term that is the term that describes the non-steady state condition, the dopamine transient, and that's the one typically that we're most interested in. So when we're using these Bayesian methods, the posterior density that we're trying to estimate is this one. We're trying to estimate the vector of model parameters shown in this model above, the parameters for the LPNT PET model. And typically, so we can construct a posterior density for each one of those parameters, but typically the one we're most interested in is gamma. So this is what it might look like for gamma. Gamma being the magnitude of the dopamine response curve. So that's what we're trying to do when we apply this method to the non-steady state problem. The other thing I really would like to highlight is what I really like about this framework is it deals with the model selection and model classification problem very, very elegantly. It's a natural fit in this framework. So when we're trying to decide, when we have a time activity curve, a single time activity curve, and we're trying to decide, is this data best fit by the LPNT PET model or by the nested MRTM model that describes only the non-steady state condition? So we're trying to decide, is there an activation or not? Which model is the most appropriate model to fit this data? Well, we can do a very simple thing. We simply expand our parameter space. We add an additional parameter, which I'm calling M for model. It's a categorical variable, so it can only take on either one of two values. And typically we'll say that if it's M equals 0, that's the MRTM model. If it's 1, it's the LPNT PET model, for example. And then you get a posterior distribution on that parameter M that looks a bit like this. So this is just for a single data set, a single time activity curve. We'll get a probability density that looks like this after performing a large number of trials. And you can see that based on a simple 50% threshold, you'll say that a certain proportion of the time model 1 LPNT PET is more likely to describe the data than model 0 MRTM. And whichever model has the highest proportion is the one that you select typically. But you can change that threshold as needed. So it works very well for doing model selection as well, and I'll show some examples in a moment of that. So I'm just going to go through some of the work that's been done in this space using these various methods. So firstly, this paper that I've already referenced from the Lyon group, Zach Iras is the first author, and they used MCMC to sample the posterior for each of the LPNT-PET model parameters. And then they applied Bayesian inference on the posterior functions that they'd estimated in order to derive their model parameter estimates. And from those, they can then construct dopamine curves. So the gamma HT term in that model that I just showed. So here's just some of the key results from that paper, just illustrating the use of this technique. So they did a number of studies, some simulation studies, but also some real data sets in cats and in humans. But in the simulated data sets, what they're showing here are the dopamine curves for a placebo condition in the top row. So we've got the LPNT-PET results. This is vintage LPNT-PET on the left with the Bayesian method on the right. And they had a number of simulated dopamine challenges of increasing magnitude as you go down the column, just showing that they achieve more reliable and more precise estimates of the dopamine curves using the Bayesian approach. And then this is just the data from their cat brain studies. This was using a serotonergic 5-HT1A receptor binding tracer, and it was a dose response study. So they're using a 5-HT1A agonist to challenge the tracer, and they saw a nice dose response relationship using their Bayesian method on the right here, with significance at each of the increasing doses. And they weren't able to see the same dose response relationship with the vintage LPNT-PET, and they had a number of quite a larger number of outliers in that data set. This is from our own group. So this paper describes the ABC, or what we call PET ABC method, and so in this paper we were trying to illustrate a wide range of uses of this method. So it included a number of simulations of 1 and 2 tissue compartment models, but also some examples of some real data sets where we were trying to analyze dopamine activation in the brains of rats. And so in this paper, we describe the use of ABC to sample the prior in the way that I showed just a little while ago, typically using a uniform prior for each of the model parameters for LPNT-PET. And then we estimate the posteriors for each model parameter in the way that I showed. And then, as in the Iras paper, we then construct dopamine curves, the gamma HT term. But because we also have the full posterior density of those parameters, we can also calculate confidence intervals on those dopamine curves. And now I'm not sure if you can hear the sound that I can hear. I forgot to check this before we started, but it doesn't matter if you don't hear the sound. So I'm just going to start these videos. This is an awake rat, as Evan mentioned. We've been doing quite a bit of this in Sydney. And I'll just let the video run. It's a awake rat. You're seeing a video from the front and the back of the micropet scanner. So it's the same animal. The animal has a marker on it attached to its forehead, which we're tracking throughout the scan. You see it move around freely in the field of view, and you can see the robot readjusting the position of the chamber so that the animal remains in the field of view. Evan, could you just give me a thumbs up if you actually heard the background noise in that video? No noise, okay. So sound not working, that's not going to be a problem. So that's the awake animal study, and this was a drug challenge study. So you can see the cannula going into a vein that leads to the jugular vein. That's where we administer both the trace, so this was C11 raclopride, and the drug, which in this case was either amphetamine or saline. So this is the amphetamine challenge result. And you're seeing an image before the drug is injected. So this is a sum image of the early part, if you like, the steady state part of the study on the left. This is a sum image of the later part of the PET scan after the drug is injected. So the non-steady state part, and you can see a clear displacement of the raclopride signal. And here we see the dopamine curves, or the dopamine curve. So this is the median of our density function on the dopamine curve. And these dashed lines indicate confidence intervals at the plus and minus 1 quartile level. And so you can see that our estimate of the dopamine curve aligns nicely with the injection of amphetamine. Below that, we're showing a trace of the motion of the animal, so as an indication of its locomotor activity. And you can see quite a dramatic change in the locomotor activity, again, coinciding with the drug injection, just to show that we've got correlation there between our motor outcome and the PET modeling. And then this is the same animal injected with the saline vehicle as a sham control. And in this case, we're getting a more or less flat dopamine curve, which is, and the baseline is fully encompassed within the confidence intervals. And as you'd expect, we see no specific change in the locomotor activity of the animal. So that's a drug challenge study. And just to show the full, more complete results, these are the time activity curves in the column on the left. So the saline in the top row, amphetamine in the bottom row. Just to show as well that with this technique, we can get confidence intervals on the time activity, the fitted time activity curves in a single subject, as well as the confidence intervals on our dopamine curve shown here. And then these are the posterior distributions, just showing that this is for gamma, just showing again that the distribution is skewed towards 0 in the saline case, and it has a nice symmetric non-zero distribution in the case of the drug challenge. So I'm going to just show you one more experiment that we've done with a similar setup and using the ABC method to analyze our results. So this was a classical or Pavlovian conditioning study. So once again, a C11 raclopride study. And the aim of this study was to determine whether we can observe endogenous dopamine release during reward-driven learning. And secondly, to investigate the relationship between dopamine release and reward prediction. So this was what the study design looked like. I won't go through this in detail, but the surgery is just for the insertion of that cannula for the jugular vein administration that I showed you. So after acclimatization, the animals were trained in a number of sessions on different days to learn to associate a particular sound with a particular reward outcome, which I'll show in a moment. And then a week later, they had each animal underwent 6 PET scans on different days. So these are the scans that each animal underwent. These are the stimuli that they learned during the training sessions and which they heard during the PET scan. So if they heard a clicking sound, they learned to expect a reward 100% of the time. If they heard a continuous tone, they expected a reward roughly half the time randomly. And if they heard white noise, they had no expectation of a reward. You can see during the PET scan, sometimes we gave them the reward after hearing the sound, sometimes we didn't. And the stimulus lasted for 2 minutes, and it occurred at 20 minutes after tracer injection. And the reward was just sugar water. And the outcome measures from this study were the PET ligand displacement, so the dopamine curves, and the number of magazine checks. So there were 2 magazines. You'll see that in the video here. So I'll just start these 2 videos now. You're not going to hear the sound, unfortunately, but take it from me. This is the conditioned stimulus where the animal is expecting a reward 100% of the time. So if I played the video, you won't be able to hear the sound, but you'll see the animal get quite excited when it hears the sound. So it's heard the sound, and now this video is just a couple of minutes later, same animal, same stimulus, and you'll see what happens when the sound finishes and the reward is delivered. So there we go, the animal gets its reward. And these are the results. So, so these are the magazine checks in the top row. So this is the same conditioned stimulus, 100% reward expectation. These are the dopamine curves with our confidence intervals, and you can see there's quite a beefy dopamine signal there, and that corresponds, this green bar indicates the time of the stimulus. And you can also see that the animal is correctly checking the magazine where there should be a reward more often than the incorrect magazine. This is the 50% conditioned stimulus where a reward is possible, but only half the time. And you still get a dopamine response, but it's a dampened response. But you can see from the confidence intervals that that we can still be fairly confident that that is a real dopamine response. And you can see the relatively poorer performance in picking the side the reward's coming from. And then this is the 0% conditioned stimulus. The animal is not checking the magazines anywhere near as often. And although LPN, well, our median from our estimate of the dopamine curve seems to indicate a dopamine, a very, very small dopamine response. But in actual fact, the confidence intervals overlap with the baseline. And so there's, we can't be confident that there was any dopamine release in that case. I'll just touch on the fact that we've also implemented this ABC method at the voxel level. So as I mentioned earlier on, it is relatively easy to parallelize this algorithm. And this was some experiments we did with Evan and his team, his postdoc Jocelyn, based on some simulations. These are the same simulated raclopride curves that Evan showed that were performed by Heather Liu. So this is a digital human brain phantom, and these green voxels in the striatum are the activated voxels. And the yellow ones are the null voxels where there's no dopamine response. So that's the truth, that's the phantom on the left. And this is our map of the results from VPET ABC, where the true positives are shown in green, true negatives in yellow. And you can see there are just a relatively small number of false negatives and just the odd false positive. Yes, false positive. So our true ratio, true prediction ratio was 89%. The false prediction ratio was 6% in this experiment. And this is the distribution of the model selection, the model classification of those voxels, just showing the full distribution of all the null voxels in the top row. And you can see that that distribution is skewed towards the MRTM model. And in the bottom row, the voxels that do contain activations are skewed towards, very heavily towards the right, which indicate the LPNT-PET model. So the last topic I want to touch on is deep learning. And so I've shown you the numerical ways we can estimate the posterior distribution. This team from MGH, led by Jin Song, have just recently published a paper showing that you can also estimate posterior distribution using deep learning. And these are the 3 deep learning networks that they designed and tested. They're all conditional variation or autoencoder variations. So they have what they call the vanilla and CVA on the left. The sections that are shown in the grey shaded area are the parts of the network that are used during testing only, and then the full network is used during training, and then the network is decoupled, and it's only the grey part that's used during the testing. And they're basically using this network this latent variable Z to estimate the posterior distribution based on this loss function. This design has the dual encoder design, and this one has, I think I've mislabeled those. So we've actually got an encoder and a decoder here, and this is the dual decoder design here on the right. And these are their estimates of the posterior distributions so this is simulated data, and they're simulating a tau binding ligand. So this is now not looking at dopamine kinetics anymore. It's looking at the steady state condition. It's a tau tracer, and these are posterior probability densities for the distribution volume ratio. And all three of them, I should mention as well that they were comparing their results with the full MC method as the gold standard. And all three variants give a pretty good estimate of the posterior distribution, but arguably the encoder, decoder, and the dual decoder perform best. So I just use that, even though that's not modeling dopamine kinetics. I use this paper as an example just to show that we can also use deep learning to estimate this posterior distribution with all the advantages in computational efficiency that you get from these methods. So I'll just wrap up here by summarizing. So the Bayesian framework, I think, is well suited to PET kinetic modeling, estimation, and model selection in general, but also specifically in the case of non-steady state models like the one we've been discussing today. It provides not only point estimates of your model parameters, but also the posterior distributions, the full posterior density distributions for those parameters from which we can make statistical inferences. And I've described three main methods for doing that, for calculating those posterior densities. MCMC, which is considered the gold standard, but has a number of drawbacks, particularly the computational efficiency and the inability to parallelize. ABC, which is an approximate method, but much more computationally efficient, and especially when you parallelize the algorithm. And we've seen that you can also use deep learning to estimate these posterior distributions. For those who are interested to try out these methods, we have made an R version of PET ABC available on GitHub. This is not a particularly fast implementation, so it's probably only suitable for analyzing ROI data, but we will be uploading the voxelized version implemented in Python very soon as well. And with that, I just want to acknowledge my collaborators, as well as our collaborators at Yale with Evan and his team, our local collaborators on the ABC work, and also our collaborators at UC Davis, ANSTO, and University of New South Wales on the mouse PET project. This is the awake animal imaging work, and also our sources of funding that have supported this work. And I will, with that, pass over to Julie to moderate the discussion section of our webinar. Thank you. I'll just stop sharing.
Thank you both, Steve and Evan. Thank you very much. If you have questions, please either raise your hand or enter your question into the chat. If there are none yet, I'll start, but please, I'll keep track of the chat. I'll start by asking Evan about the generalizability of the LP&T PET method in terms of the extent to which it can be used or has been applied to track other transient phenomena that occur during a PET scan?
Yeah, thanks. It's a great question. So most of the work so far has been on dopamine, with a little bit starting to seep into the examination of other transient responses of neurotransmitters or drugs for that matter. But the critical thing, I think, to understand is that the choice of tracer is very important. And why? Because we're measuring, of course, dopamine indirectly, or any other neurotransmitter or any other phenomenon. The other, the endogenous chemicals are not radioactive. So you're relying very heavily on the responsivity of the tracer. And in point of fact, that ends up being fast enough dissociation of the tracer to respond to the increase in competition within the time window of the scan.
What about estimates of timing of the endogenous neurotransmitters? I mean, other examples being serotonin or other endogenous transmitters, applicability to opioids. I remember early studies with buprenorphine.
That's interesting. And I'm not familiar with the particular need for knowledge of timing. I can tell you that what we've been motivated by, which again takes us back to dopamine, is the now kind of old claim by Volkoff and Swenson, suggesting that dopamine itself and its transient behavior encodes reward. And so the question that we have been asking in part is, is there a value in knowing that transient in all of its details? And I think on top of that, but again, it's mostly a dopamine thing, the action of some drugs has been hypothesized to be related to their ability to alter that time course of dopamine, and hence time course of the feeling of reward. So that was kind of a dodge.
Just one quick extension of that, and that's in terms of the context of timing of an exogenous drug in the brain. Are there parallels? What is your thought about the performance? Its limitations.
I think, at the risk of sounding like a broken record, I'd say that ultimately the response of the whole system, the Achilles heel, is how fast the tracer, whatever it is, if we're setting up a system of competition, how fast will the tracer respond? Certainly, there are drugs that were developed because they work faster or slower than the first one in the market. I'm thinking of breviracetam and leviracetam, which were developed to treat epilepsy, I think, originally. So you might want to know why these things are fast or slow. I think Wobao was asking, to sort of complement your question, what's the resolution of all this? And it's a tricky question. It's not the same, of course, as your frame time. So, your binning, but it is a question of how close in time can we differentiate one, let's say, peak in the dopamine curve from another. And I think we're talking about, a few minutes.
If there's nothing else on that topic, then I would like to ask Dr. Golden to please pose his question.
Thank you very much. Sorry, Evan, I missed your talk, but I was listening to Stephen. I mean, great, very interesting topic. I just saw from Steve's talk that the results were based on animal. So I was just wondering, have you done PET study for dopamine reward learning in human? And what are the challenges with these studies?
Yeah, thanks for your question. So I think I'm going to pass this one actually over to my colleague Evan. The short answer is that for us, we have not used these methods in humans so far, and unfortunately, we're unlikely to do that in the near future because we no longer have access to C11 raclopride in Sydney. We do have a very nice high sensitivity total body PET system, and it would have been lovely to. I think actually we could potentially address some of the questions that Bale was asking around the importance of high temporal resolution, because I think with the high sensitivity, there's the potential to extract more nuanced information about the timing of these dopamine signals with these high sensitivity PET scanners. But unfortunately, we no longer have access to C11 raclopride for a variety of reasons. But I do know Evan has done a lot of this type of work, and we've looked at some of their data as well. So Evan, do you want to talk about your dopamine studies just briefly, even though you covered that in your talk?
Sure, and I'll just say for everybody that came late, it is, for good or ill, it's being recorded, so they can go back and listen.
That's great.
Yeah, so we have done in our group smoking in the scanner and smoking under different treatment conditions to look for alterations in timing of the dopamine response. And our colleagues led by Ansel Hilmer and Kelly Cosgrove have also looked at smoking marijuana in the scanner and detected dopamine responses. And I would say, if I were being completely honest, probably not a good thing to do on the internet, that one of the remaining challenges, other than just addressing false negatives and false positives, which is about modeling and image processing, is what to do about transient motion. And is there transient motion when somebody smokes that is or could be confused with a bona fide dopamine response? And I'd say that's something else that we are contemplating addressing once and for all, because it's always in the back of my mind.
Thank you very much.
Okay, we have a few more minutes. Please raise your hand or submit your question. I'll try to find your hand and read the question. I'll ask Steve a question about the ABC, general ABC approach. And maybe you can just summarize for us again what you think the real strengths are of the estimation of the posterior distribution of the model parameters, and what additionally that might offer when we are really just interested in just the parameter estimates, and what you think the ABC additionally offers.
Yeah, sure. Thanks, Julie. So I think for me, the main thing it offers is more complete information about the reliability of our parameter estimates. So when we do model fitting using conventional least squares method, we get our point estimates. We might be able to get an estimate of the variance in that estimate. But there is an underlying assumption about the error distribution. We typically assume that we have normally distributed estimates. With methods like ABC, I like the fact that you can see the posterior density distribution. You see the full distribution. You can see whether it's symmetric, whether it looks like it is normally distributed. Sometimes we see bimodal distributions, which don't lend themselves to traditional methods of error evaluation. So there's a lot of useful information in those posterior distributions about how much certainty we can have about the parameter estimates that we get. And I like the fact that we get that in single subject data as well. So you don't need to do a group analysis in order to get that information about the reliability of the parameter estimates. There's a whole lot of other things you can do with those posterior density functions that I didn't describe in my talk, and we haven't really delved into this much, but you can start to look as well at correlations between your parameters in single subject data and how they're covariing. You can look at joint conditional probabilities of these different parameters. So I think there's a lot of rich information there, and we're only just starting to look into that. But for me, the main thing is just a more complete picture of how reliable our parameter estimates are.
Well, thank you. I would think it would be of interest to look at covariance and other factors like that, and just examine some underlying assumptions of the method too.
Yes. Sorry, Julie, I was just going to say, just to add one more point, another area that we're really interested in, I think, is also spatial correlations. So we're starting to apply this ABC method on a voxel by voxel basis. And of course, at the moment, we're still treating those voxel time activity curves as independent measures. But we know there are spatial correlations and there are clusters there. And I think it would be really interesting to look at not only temporal not only correlations between the parameters, but also across time, but also correlations in the spatial domain as well.
Wonderful. Thank you. Christian Schmitz has a question for Steve about the temporal resolutions in your experiments on conditioning in animals. Classical electrophysiological experiments show dopaminergic dynamics on a time scale of sub-seconds to seconds. How do you interpret the dynamics of seconds to minutes derived by your model?
Yeah, so we obviously don't have the temporal resolution of electrophysiology, and but I mean the literature suggests that these dopamine reward signals are happening not really on the sub-second scale, but it's more like over several seconds. I think where with the temporal resolution we have in our conventional PET systems, especially the animal PET systems, we're right at the limits of what we can really detect in terms of dopamine timing with these methods. But I do think with these high sensitivity human and animal PET systems, but particularly the human systems, I think we're going to be able to extend those limits. I think we can extract, as I hinted earlier on, I think we can extract more nuanced information at a higher temporal scale, and that's something we'd be really keen to explore. But certainly using our current methods, and particularly our animal scanners, we can't compete in terms of the temporal resolution with what's achievable with electrophysiology.
Thank you, Steve. Terry Jones.
A question a question for Evan. As you know, Evan, there's a pandemic now of addiction for gaming. And people are getting very worried about teenagers and even younger than teenagers getting addicted. Now, we've got very high sensitive PET scanner, as you know. When are you going to move away from smoking and begin to look at this pandemic with your systems?
Yeah, I think it's a wonderful idea. And if I'm not mistaken, you were in on that original gaming paper. So we're sort of returning to an important question, but maybe with higher temporal resolution and a better model, I'd be delighted to try it. Can I sort of pivot off of Terry's question to give our group and Rich Carson, who developed with his whole team at multiple sites, the Neuro Explorer, which is one of the high sensitivity PET scanners that Steve is alluding to. We have it here at Yale. And to say that Heather Liu, who was my graduate student a few years ago, did a simulation study, and she asked the question, we're connected a little bit to Terry's question, a little bit to Steve's statement. If we had this wonderful higher sensitivity, 10x in fact, improved sensitivity in PET scanners, how much better would our ability be to detect dopamine curves, such as what we saw or what we showed in our simulations? And it was quite striking. So now that we actually have the scanner in place, we're gearing up to try to do some studies. Maybe they would be the smoking studies all over again to see if our predictions were right of improved ability to detect and distinguish, to go to Guobang's question, between dopamine curves that peak at different times. However, I have to admit that squeezing somebody in there with their cigarette could be a bit tricky. So maybe I'm going to have to go back to Terry and find out, could we hook up a gaming system instead, which would cause what we expect to be a transient dopamine effect, but without having to have somebody bringing their hand to their mouth and banging their cigarette into the insides of the PET scanner.
Well, as people are less smoking now, but more and more people are getting addicted to games, that's where the big investigation is needed, I think, particularly in young people. And we can drop the dose with the high sensitivity systems and, begin to ethically look at this.
Yeah, I think it's a great idea. And as you also say, a lot of what has limited us for years in studying teenagers is the dose, but we may be able to reduce that sufficiently to be, for it to be acceptable ethically. Good point.
Very interesting. I guess we're sort of wrapping up. Guobao asked whether it's feasible, Steve, or is it feasible or with any challenge to run ABC at the pixel level, such as based on the total body dynamics? So extending into your realm, the total body dynamics.
Yeah. Thanks for that question, Guobao. I did hint at this earlier on, but look, I think it's very feasible to run this algorithm at the voxel level, even when you have many hundreds of thousands of voxels, as in total body PET. So actually, only just yesterday, I have a student who's been working on this on the voxelized version, and he only yesterday told me that he's now achieved another significant speed up factor. What he's done is there's two main things. You can take a lot of the iterative the simulations out of any voxel loop, so you can do all your simulations up front, do 1 million or more of those simulations, and then you can draw from those samples on a voxel by voxel level. And the second thing he did was to vectorize the algorithm, and that was very successful. So using the vectorization in the NumPy library, and when he did that, he got quite a significant speed up. So just to throw some really quick numbers at it, he was telling me it took on our I can't remember all the details, but it's on our high performance computer. But just using a single CPU, he was able to process about 2,000 voxels in 2 minutes. So that's not unrealistic to scale that up for a total body dynamic PET study, I think.
Thank you. And thank you for the comments in the chat. I think Ginny has a question.
Thank you, sir. Thank you, Evan and Steve. Wonderful talk. Just a follow up what Steve mentioned about voxelization. I just wondering, so when you do this voxelized analysis, will there any benefit to consider the correlation between the voxels, or it doesn't matter when you do this ABC or MCMC calculation?
Yeah, I think there is. I touched on that just briefly earlier on. We haven't tried to look at that just yet. We're just treating each voxel time activity curve as an independent measure. But I think there's a lot of benefit in looking at the correlations between, particularly not just neighbouring voxels. I think also looking at correlations across big distances as well, particularly in total body PET. I think these Bayesian methods can accommodate that sort of analysis. It's not something we've done yet, but we're certainly keen to do that. I think that the framework allows you to look at correlations between parameters and between kinetics across voxels. So yeah, definitely worthwhile investigating, I think.
Yeah, I think we're about wrapping up. I would just like to thank everyone, and I'm going to turn it over to Guobao and remind everyone about neuroreceptor mapping 2024, the note in the chat.